Thank you, Dushan, for this introduction. I would like also to extend my thank you to the science coordinators, Chris and Kirsten, as well for their kind introduction and invitation. I would also like to thank Heather for helping with the logistics, as well as the rest of the session organizers. So I'm really excited to be back in Richland today to share with you how we are using spatial metabolomics in addition to other analytical tools to better understand how organisms respond to changing environmental conditions down to the micron scale. Now, before I do that, I would like to take a moment to acknowledge all the EMSL staff that's been detrimental in assisting myself and my team in past and current user proposals, even with project management, sample handling, data collection, as well as their effort in sample processing and data interpretation. Thank you all for your dedicated efforts. None of the stuff that I'll be showing today would be possible without your help. Second, I would like to acknowledge my lab members as well for their hard work. The data that I'll be showing today was actually collected by two former lab members, Dr. Linaya Honecker, who was a postdoc at the University of Arizona, co-advised by myself and Dr. Laura Meredith, and she's currently at Lawrence Berkeley National Lab. Gina Hildebrand was also a master's student in my lab who did a fantastic job on this project. So now, as you all know, climate change is expected to increase abiotic stresses on different ecosystems. Now, we also know that soil microbes and plants play a significant role in driving ecosystem processes. So any change in climate weather events is expected to influence biogeochemical cycling that's taking place by microbes and plant communities. To be able to gain a comprehensive understanding of how ecosystem function and processes will change as a function of changing environmental conditions, it's essential to use a dual approach. One that really looks at the combined reactions at the ecosystem scale, but at the same time, it really hones down into what's happening at the organism level. Understanding what happens at the organism's level is really important if we were to understand ecosystem processes. So luckily to us, spatial metabolomics made possible through imaging mass spec allows us to understand what happens at the organism down to the micron scale. By understanding how the organisms change their metabolic processes due to changing environmental conditions is really important for us if we were to understand ecosystem scale processes. By using spatial metabolomics, researchers are able to uncover hidden patterns, mechanisms, and adaptation strategies by different organisms that might not be possible by using different analytical tools. Also, it provides insights into both the localized and the wide ecosystem responses within any given ecosystem. So using spatial metabolomics allows us to understand these patterns down to the micron scale, which is really important for applications in ecology, agriculture, biotechnology, and health research. What I'm going to be doing today is giving you an example of how we are using spatial metabolomics in combination with other analytical tools such as liquid state NMR, FTIC-RMS, and nanosims to understand how organisms respond to changing environmental conditions. In particular, we'll be looking at drought as one of the potential environmental changes with climate change. Oh, this, all right. All right, we got it. All right. So back in 2019, we conducted a 65-day drought experiment at the tropical rainforest at Biosphere 2, part of the WALT campaign in collaboration with Chris Werner from the University of Freiburg in Germany and Dr. Laura Meredith from the University of Arizona. The main goal of this WALT campaign was to track fully from genes to ecosystem the processes that drive the cycling of carbon and water in tropical rainforest under a drought. So to conduct this experiment, we imposed a 9.5-week drought on this system, and we pumped labeled CO2 into the atmosphere at 2 different time points, before drought and during drought. And the main goal of pumping labeled CO2 into the atmosphere was to track how the ecosystem responds to these changing environmental conditions, in particular a drought. Now, ecosystem-wide changes in carbon fluxes was published in 2021 in Science, in case if you're interested in reading more about this experiment. But briefly, what we observed was a decrease in the carbon emissions above ground, as well as carbon transport below ground that was consistent with the increase of the severity of the drought. While I don't necessarily have time to talk about the full details of the ecosystem-wide responses, I'll be mostly focusing on our other experiment where we wanted to understand some specifics within that individual scale organization. So as I mentioned, ecosystem scale observation was a decrease in the carbon flux above ground and carbon transport below ground. While this observation might not be too surprising to many, what was interesting was the observation that that ecosystem response was dependent on the plant functional groups. For example, canopy plants dominated ecosystem flux, whereas understory plant responded differently. So to be able to better understand how these individual plants are responding to this drought, we carried out another labeling experiment that was also performed before drought and during drought. And the main goal of this labeling experiment was to understand carbon allocation within individual plants. Our aim was to gain a deeper understanding into ecosystem response by examining the distinctive behavior of individual plants, because that in response of each individual plant is what gives rise to that ecosystem response. As well as we wanted to see if this response of individual plant to drought will also influence plant microbe interaction at the rhizosphere interface, which is mediated by roots. Now, to be able to do this experiment, what we've done, we chose 3 different tropical rainforest plants. We're going to be looking at piper, hibiscus, and clitoria. These 3 plants were chosen because they are dominant tropical rainforest plants. Plus also they represent different understory and canopy plants. We have piper and hibiscus are both understory plants, whereas clitoria is a canopy plant. At the same time, clitoria is a legume plant compared to hibiscus and piper. And since legumes are known to associate with microbial communities, we wanted to see also if a plant that is able to undergo symbiotic relationship with microbes will respond differently to drought compared to hibiscus and piper. So the way we did this experiment is that we gently dug the roots of these plants without disconnecting the roots from the plant. This whole experiment was done directly in the tropical rainforest without removing the plants from the tropical rainforest, because we wanted to really look at the effect of drought as natural as possible. So we gently dug the roots of the plants from below ground, and then we gave the roots either labeled pyruvate at the C1 position or at the C2 position. The reason we gave the plants pyruvate was to see how the plant will metabolize pyruvate under ambient conditions versus under stressful or drought conditions. Also, we chose labeled pyruvate at the C1 position to be able to track how that pyruvate is metabolized into CO2, because from previous work, it's been shown that the C1 on the pyruvate is used by the plant to produce CO2, whereas the second carbon on the pyruvate is used by the plant to produce downstream metabolites and eventually VOC. So by giving the plant pyruvate at the C1 or at the C2 position, we'll be able to track how this plant responds to the drought by how it's producing CO2 above the ground, as well as how it's producing root exudates below ground. And by understanding that individual plant, then we could understand how it responds to that drought and how this gives rise to that ecosystem response. Now, in addition to giving the plants C1 and C2 labeled pyruvate, we also took half of the roots and washed them and part of the roots were washed and the remaining part remained unwashed. And the reason we washed part of the root is because we wanted to see if the microbial communities living on the surface of the root will play a role in how the plants will respond to these drought conditions, especially since we have a legume plant and we know legumes like to associate with microbial communities. So now we have multiple treatments going on. We have 3 different plant types. We have C1 and C2 labeled pyruvate. We have washed, unwashed roots, and we have before drought and during drought. And we wanted to use all that data to really understand how the plants respond to the drought, if we see difference between the plant roots, and if we see difference between whether the roots were washed versus unwashed. So as you could see here in the image, we were able to put the roots within a tube to be able to measure how much CO2 is coming off the roots, as well as how much VOC, because again, this contributes to the ecosystem's wide response. In addition to tracking those emissions from the roots, we also decided to look internally about what's happening within the root structure. To do that, we used multiple analytical tools. The first one was liquid state NMR in collaboration with Dave Hoyt here at EMSL and his team. And the reason we decided to use liquid state NMR because we wanted to look at the bulk change in smaller molecular weight metabolites within the root. Also, NMR is really good in tracking labeled metabolite. So this way, we could see whether the roots are metabolizing pyruvate differently under ambient versus drought conditions, and we will be able to see which byproducts were produced as a result of pyruvate metabolism. In addition to doing liquid state NMR, we also looked at the spatial distribution of the metabolites within the root structure in collaboration with Dushin and his team. Now, this multi-mass spec was done on a 15 Tesla FTICR MS, in addition to allowing us to look at where these metabolites are within the root structure. It's also complementary to the NMR because it allows us to look at relatively larger molecular weight compounds than what we detected with NMR. And finally, we use nanosims in collaboration with John Cliff and his team to be able to look at the spatial distribution of the labeled metabolites within the root structure. So the majority of my talk today will be focused on the results we got by integrating 3 analytical tools here at EMSL. Now, as I mentioned earlier, we wanted to understand how the plant will respond to the drought. So that's why we are investigating the root structure, because we know that the root connects above ground and below ground. To be able to understand how the changes happening within the plant root will influence rhizosphere, we also collected the soil that is part of the unwashed roots. We then looked at the metabolic profile of the rhizosphere soil in collaboration with Rosary Chu and Jason Toyoda using FTICR MS to look at those relatively large molecular weight compounds. And also we did NMR with Dave Hoyt and his team to look at the small molecular weight compounds. So by using both FTICR and NMR, we are able to provide a comprehensive overview of the type of metabolite present in the roots, in the soil. And we wanted to compare them to the type of metabolites present in the root to be able to see how the root dynamics are influencing metabolites in the rhizosphere. To also see if the changes in the root dynamics will influence metabolite in the rhizosphere and how this will influence microbial communities, we also looked at the structure and the composition of the microbial communities using amplicon sequencing at the University of Arizona. All right, so now moving on to the data. First, I'll be showing you, missing slide here. Sorry. Yes, maybe I'll just go over it. Okay. So what I'm going to show you first was a PCA analysis of the NMR data of the metabolites present within the root structure. What we've done first were to put all the data collectively in one PCA analysis. As I mentioned, we have a lot of treatments going on. We have the effect of drought, we have the effect of different plant functional groups, and we also have the effect of whether the plants were washed or not. When we first put all the data together on a PCA analysis, what we observed was that the major difference in the metabolic profile of the root was actually driven by the plant functional group. It turned out that the type of plant will dictate the type of metabolite that we see within the root structure. So now we know that the different functional groups dictate the type of metabolites present in the root structure. We then moved on to look at each plant individually to be able to look at the effect of the drought and whether the roots were washed versus unwashed. So this is what I have on this slide. Now we're looking at each plant individually. We have the piper here, we have a hibiscus here, and we have clitoria. Now, each dot on the figure represents an individual sample, and the shapes indicate whether these were washed versus unwashed, and the colors within each figure represent whether they are under drought or pre-drought condition. So let's start first by looking at the top two panels. We have piper and hibiscus. If you look at these two PCA analyses, you could see that for these two plants, drought was the largest driver of changes in the plant metabolome. For example, these 4 dots represent samples that were collected under drought condition, regardless of whether they were washed versus unwashed, and the same thing for piper, suggesting that both for piper and hibiscus, they are really sensitive to drought conditions, and the plant root changed its metabolic profile based on these conditions. Now, if we look closely in this figure, we see that during drought, we see an overall enrichment in sugars, amino acids, and betaine that was observed for both hibiscus and piper. These kind of metabolites are known to be osmolytes. So what the plants are doing, they are upregulating the production of amino acids, sugars, and betaine. These are osmolytes to prevent water loss and to help the plant during these stressful conditions. Also, because we use labeled pyruvate, using NMR, we were able to also track labeled pyruvate as it was metabolized by the plant root into different substrate. Overall, we saw we have less carbon-13 incorporation under drought. The plants are really stressed and they stopped producing biomass, as well as they diverted their carbon allocation away from energy production and TCA cycle. Again, when the plants are stressed, they're actually changing their metabolic pathway to prevent biomass production, to prevent wasting energy, but rather they are upregulating osmolytes to help the plant during these stressful conditions. In contrast, when we actually looked at clitoria, which again, I mentioned clitoria is a legume plant, if you look at this PCA figure, the separation between the dots is based on whether the plant root was washed or not. It seems that clitoria, even though we see some indication that the metabolic profile was changing due to drought conditions, the majority of the shift in the metabolic composition was driven whether the plant roots were washed or not, suggesting that the presence of microbial communities on the top of the root structure is actually changing or helping the plant metabolize pyruvate. And I'm going to be talking about this a little bit more later on. So just kind of to summarize this slide, out of the 3 different plant species, piper and hibiscus appear to be really sensitive to drought. They change the metabolism internally to the root to help with these stressful conditions, whereas clitoria, on the other hand, was mostly impacted by whether the microbial communities on the top of the root structure were present or not. And it seems like there is some association between the plant root and the microbe that is helping the plant overcome these stressful conditions. Now, to better understand how these metabolites are spatially distributed within the root structure, then we moved on to do spatial metabolomics using MALDI mass spec. So what we've done with this was to take small cross-section, transverse cross-section of the root, allowing us to look at the internal structure of the root. Here I'm showing microscopy images of the 3 different roots. And you can see within these roots, there's different structures within the root. I'll be going over the different root structures, the importance of these structures, and then we're going to see how drought changed how the plants redistribute metabolites within this root structure. So first, I want to point your attention to the vasculature, which is in the middle of this root structure. Vasculature is known to connect all internal organs of the plant from the root to the stem to the leaves. Therefore, the main goal for the vasculature within the plant root is to transport water, nutrients, and all needed metabolites throughout the plant body. In addition to the vasculature, we have the epidermis, which is the outer layer structure of the root. It protects the root's internal tissues from pathogens, disease. It also is known as a gatekeeper to protect the nutrients inside the root and to prevent any loss to the outside environment. And finally, we have the cortex, which is the layer in between the vasculature and epidermis. The major role of the cortex is to act as a storage system for the entire plant, where carbon sources can be accessed when needed. Now, to be able to now see how the different metabolites were allocated differently between these three structures, we use MALDI mass spec, which was done on a 15 Tesla FTIC-RMS. As I mentioned earlier, by doing this approach, we're going to look at how specific metabolites are localized within this root structure, but also it is complementary to NMR because it allows us to look at relatively larger molecular weight compounds. This is an example of one metabolite that was present within the three different roots. Here, what I am showing is, again, one particular metabolite, and the color indicates how abundant that metabolite is within the root structure. With the yellower the color, the more abundant that metabolite. If you look at Piper, you could see that this metabolite is present in the vasculature as well as in the epidermis of the plant. The same thing for hibiscus, whereas for clitoria, this metabolite is mostly abundant in the middle of this plant root. So what we've done next was to manually go through every single metabolite we identified with MALDI mass spec. We then assigned whether that metabolite was present in the cortex, vasculature, and epidermis. We counted how many times we observed that metabolite before drought, during drought, and then we assigned a class of that metabolite to see what kind of metabolite is present at each treatment and within which structure within this root. So first, I'll be showing you the results from Piper. Just a summary for Piper. Piper was really sensitive to drought. Using NMR, we see upregulation of sugars and amino acids. We saw diversion of energy from biomass production to more increase the fitness of the root to prevent any water loss. So now we're going to look at the MALDI result to see first if they are consistent with what we observed with the NMR, but also to see if we could find other mechanisms of adaptation against these stressful conditions. So I'm showing the data in two different ways. On the left here, I'm showing a bar diagram of how many metabolites were present in the cortex or the vasculature of the plant during drought and pre-drought condition. And on the right, I'm showing few examples of how the MALDI images looked like for two given metabolites. So first, I want to first look at the sugars. As you could see, under drought conditions, we see a significant increase in the number of metabolites that are assigned to be sugars. And this is an example of the MALDI image where this given sugar was not necessarily abundant in the cortex before drought. However, its abundance increased significantly during drought condition. These results are actually consistent with the NMR data. With the NMR, we show upregulation of sugars, and using MALDI MS, we were able to see that this root structure is upregulating the production of sugars and is actually storing it in the cortex. As I mentioned earlier, the cortex is used for storage of nutrients. So what the plant root is doing is really investing in production of sugar and storing that sugar in the cortex so that the plant will have access to it as needed. In addition to sugar, you could also see an increase in the amino acids as well in the cortex, which again consistent with the role of the cortex of storing nutrients for the plant root to use as needed, especially under stressful conditions. What was also interesting looking at the MALDI MS is that we saw upregulation of lignin-like compounds, especially in the epidermis. As I mentioned, epidermis is the outer layer of the root structure. It really helps increase the rigidity of that root and prevent water loss. using MALDI-MS, we saw evidence that this particular plant is upregulating the production of lignin to actually strengthen its outermost layer. To start with, Piper actually have a very thin outer layer membrane. So this plant, what's doing is actually increasing the production of lignin. That is what we call structural defense, to reinforce its cell wall, increase the mechanistic ability, and reducing water leakage. In addition to lignin, we also saw increase in the condensed aromatic. So by combining NMR and MALDI-MS, we saw evidence that Piper is really sensitive, and it's using two strategies to avoid that stressful conditions, increasing nutrients in the cortex, but also lignifying its cell wall to prevent any water loss. Now, moving on to hibiscus. Again, a quick review of hibiscus. Hibiscus behaved similar to Piper. It upregulated sugars and amino acids, as we've seen with NMR. So now we're going to look at the MALDI-MS to see if we see evidence for that, plus if there is any additional adaptation strategies that is different than what we observed with Piper. If we first look at the carbohydrates, we also see a similar observation to what we observed in the NMR data, as well as what we observed with Piper. We see an upregulation of sugar as in the cortex, where the cortex actually stores the nutrients, again, consistent with the NMR data. However, we did not see upregulation of lignin structure. For Piper, since it has a thin cell wall, the plant really lignified its cell wall. Whereas with hibiscus, to start with, hibiscus already have a very strong cell and a thick cell wall. So hibiscus decided not to really lignify its cell wall because it's already lignified to start with, but rather it invested in different strategies. In particular, we saw evidence of increase in antioxidants, in particular ascorbic acid. Usually ascorbic acid is a tannin-like compound that is capable of increasing water use efficiency in drought. In addition to producing antioxidants, we also saw increase in the lipid-like compounds in the cortex. Lipids are actually known to increase the cell membrane viscosity to support water turgor. So in other words, while hibiscus increased sugar and amino acids similar to what we observed in Piper, it used a different strategy to avoid stress by increasing antioxidants and lipid production because it already have a strong cell wall. But overall, both Piper and hibiscus appear to be really sensitive to drought, and therefore they restructured the metabolome internally to kind of overcome these stressful conditions. Now we're going to move on to clitoria. As we've seen with the NMR data, the majority of the change in the metabolome or the bulk metabolome was due to whether the plant was washed or not. While we saw some evidence of change in the metabolome due to drought conditions, it was mostly influenced by whether that root was washed or not. When we actually looked at the MALDI images here shown on the left, there was something really interesting when we went through the metabolites one by one. We observed, this is an example of one of the sugar metabolites we observed for MALDI, where we saw localized and asymmetrical metabolic distribution around a region that we assume it was a nodule present within the epidermal and the cortical structure of the unwashed roots compared to the washed roots where it was completely missing. This provides further evidence that clitoria really benefits from that plant microbe interaction, and this could be an evidence that there was a root structure, and this plant is really directing all its sugars to where the nodule is to be able to communicate with the microbe, and the microbes in return will help the plant during these stressful conditions. You could see another microscopy image here where I'm showing part of the epidermal wall outlined here in white, and here in black is where we hypothesize the nodule cells were present. So kind of just review that, MALDI imaging allowed us to see evidence of some interactions occurring between the plant and microbe, and we hypothesize that this interaction between the plant and the microbe is why the internal metabolome using NMR was not changing, because the microbes are actually helping the plant overcome these stressful conditions. To further confirm this, we also used nanosims in collaboration with John and his team. Here I'm showing the nanosim images only for clitoria, where we have the unwashed on the bottom and the washed on the top. And wherever you have that pinkish color indicates where we have an isotopically labeled metabolite. Remember, we used labeled pyruvate, and labeled pyruvate will be metabolized by the plant into different substrates. Before the plant was washed, we observed some really localized distribution of that, of a given metabolite, which we hypothesize here it is a specific sugar that was produced by the plant as a result of pyruvate metabolism. And it's localized because, again, this plant have interactions with the microbe, and it could be this is where potentially the nodule was present. However, when we wash the roots, we see a different distribution of the labeled metabolite, where now we have the distribution is all over that root, suggesting that by washing that root, we might have disrupted that interaction between the microbe and the plant. And now the plant is not necessarily allocating specific metabolite within a specific structure of the root, but rather than it's spread all over the place. So kind of to sum up this internal metabolome story, both Piper and hibiscus were really sensitive to drought. They changed the internal metabolome structure. They stopped investing in increasing their biomass. We also saw a decrease in CO2 production above ground. Whereas with clitoria, it seems that clitoria is really benefiting from plant microbe interaction to help this plant overcome a drought. Now, to be able to further take this further and understand how the change in the internal structure within the root is influencing the rhizosphere processes, we then, as I mentioned earlier, we took the rhizosphere soil from the unwashed roots, and we looked at the metabolic profile as well as the microbial communities. In the remaining minutes, I'm going to show you how the metabolome profile of the rhizosphere changed with drought, how the microbial community changed with drought, and then finally we wanted to see whether the metabolome of the root will dictate what happens in the rhizosphere. So here what I'm showing is a PCA analysis of the FTICR data on the left and the amplicon sequencing or 16S data, or looking at the bacterial structure here on the right. Again, each dot here represents a specific sample, and the colors represent the different plant species, whereas the shape represents whether it's under drought or pre-drought condition. We're going to start first by looking at the FTICR data. Again, FTICR data is complementary to NMR, allowing us to look at relatively larger molecular weight compounds compared to what we see with the NMR. As you could look at this figure, and we have some statistics shown here in this table, we see that both the species and the treatment are impacting the change in the metabolome profile in the rhizosphere. So first, let's start with the species. We've seen that depending on the kind of the species, the type of metabolites in the root will differ. Also, we're seeing here that the kind of the metabolites in the rhizosphere is also dictated by the type of the functional group we're using, suggesting that whatever metabolites are being produced in the root will directly influence the rhizosphere dynamics. Second, we also see a significant impact of the drought, suggesting that there's two things at play in the rhizosphere. It could be that the changes in the root metabolome will directly influence the metabolome in the rhizosphere, and that's why we are seeing an effect of drought, because we've seen from the internal analysis that the root metabolome changed with drought. So if the rhizosphere is directly influenced by the root metabolome, any changes in the root metabolome under drought will then be influencing the root metabolome in the rhizosphere. Also, it could be that the rhizosphere metabolome is influenced by microbial communities. We know that microbes live in the rhizosphere, and if the microbes are being impacted by drought, we should see that impact in the rhizosphere. And it could be also the combination of both. So when we looked at the distribution of the microbial communities in the rhizosphere, we see that mostly what's driving the change in the microbial communities in the rhizosphere is actually the plant functional group, rather than drought. So what this tells us is that the microbial communities actually in the rhizosphere are directly shaped by the root metabolome, and they are not being influenced directly by drought, but rather whatever happening in the root metabolome is influencing the rhizosphere metabolome, and the change in the rhizosphere metabolome is actually influencing the type of microbial community in the rhizosphere. So in other words, we are saying that the root metabolome is both shaping the rhizosphere metabolome as well as the microbial communities living in the rhizosphere. So now I'm going to show you two final figures, one of the FTICR data and how we are using the FTICR data to understand the dynamics of the rhizosphere metabolome. And then I'm going to show you some data on the amplicon sequencing of the rhizosphere microbiome. So here I'm showing Van Krevelen diagrams of the three different plants. We have Piper, hibiscus, and clitoria. Again, I am looking here at the rhizosphere. What I want you to focus is on the green dots here in the Piper. Green dots represent metabolites that were present only during pre-drought conditions, whereas in brown are metabolites that were present after or during drought. So for Piper, as I mentioned, Piper completely shut down. It really stopped investing in biomass, in producing CO2, and rather it really invested in really increasing the rigidity of its metabolome within the root structure. Using label 13C, we used the NMR data to track how much exudates were released into the rhizosphere. And what we observed was really a small amount of exudates were released when Piper was under stress. Using FTICR data, also, we see that we do not have much production. So in brown should be all the metabolites that are present in the rhizosphere during drought. We don't see much production of metabolites during drought compared, for example, to clitoria. In particular, we see a really decrease in the amount of sugars and lignins in the rhizosphere soil during drought compared to pre-drought. And this is consistent with the fact that the Piper is really keeping all its sugar and lignin within the root structure rather than releasing it into the atmosphere to be able to overcome these stressful conditions. Clitoria, on the other hand, when we looked at the root structure, we did not see a significant shift in the metabolome using NMR. When we looked at the MALDI-MS, we saw the most changes are actually due to whether the microbes were present or not. When we looked at the FTICR results, we saw a significant production or release of phenolic compounds. These are secondary metabolites that are actually produced to attract microbial communities. So what clitoria is doing, rather than investing energy in restructuring the metabolome of its root, what it's doing, it's actually producing and releasing a lot of phenolic compounds to attract more microbial communities to help the plant overcome these stressful conditions without the need to do anything to its root structure. Hibiscus was actually in between Piper and clitoria. Finally, when we looked at the microbial communities, we saw further evidence that clitoria changed its metabolism not internally, but rather externally to attract more organisms to actually help the plant overcome a drought. Here is a dye Venn diagram. We were looking at the microbial communities present in each species. Here we have the hibiscus, clitoria, and Piper. And this dye Venn diagram allows you to look at which organisms are unique to each plant and which are shared within each plant. But what I want you to focus on is which organisms are actually changing under drought conditions. When we look at clitoria, we actually see around 27 ASV got depleted under drought with enrichment of 14, whereas with Piper, we do not see a significant change in the number, further showing that Piper does not really care about the microbial communities outside, it's just trying to really maintain its fitness internally, whereas with clitoria, rather than investing in changing the root structure, it's actually changing its exterior metabolome in the rhizosphere to control which organism it will attract to actually help it overcome the stress. In particular, if we look at which taxa were actually enriched, we see evidence of actinobacteria and chlorophyxia that are actually upregulated during drought conditions. Both of these organisms are actually known to be present in environments that are lack of nutrients or environment that are lack of water. We call these organisms are really a desiccant resistant organism because they're actually able to be present in an environment that do not have water. To kind of quickly summarize all what I talked about, Hopefully, you could see today how we are actually looking or using spatial metabolomics in addition to NMR, FTICR, and nanosims to understand what happens within the plant as an individual organism and how that response at the individual scale gives rise to that ecosystem scale. Without understanding what's happening at that micron scale, we won't be able to come up with mitigation strategies or help the organ or help the system overcome these stressful conditions. So, as I mentioned, that Clitoria really increased its microbial association. To prove that, we did some microbe metabolite co-occurrence networks. And if you look at the figure on the top, this is how the microbial interaction looked for Clitoria before drought. However, after drought, or usually the phase during drought, we see an increase in the hubs of that network where we see a significant microbe metabolite interaction. This is an example of hub 3. This is how hub 3 looked like before drought. We basically just have metabolites interacting together. The same hub changed during drought, where we have now a strong association between microbes and metabolite, again, further showing us how each plant used different strategies with the main goal is to increase that fitness. And it's really important to look at each plant individually if we were to understand that ecosystem response. With that, I want to thank all of you for your attendance, as well as another thank you for EMSL staff. None of the data that I collected today would be possible without all your help, as well as the lab members and all the funding agency that made this possible. Thank you.
Oh, this was really great and inspiring talk. Yeah. Thanks, Malak. This is really great example how we can use multiple EMSL capabilities to actually combine them. They're complementary. You can see that they're not overlapped. They're really complementary. Then we can address really important biological questions, as Malak really great story. So we're open for questions now. So yeah, please, if you have here or I don't know online as well, we can go after if. Yeah, okay.
Again. I was saying that you're mentioning about the washing the roots. Did you sterilize the roots? Did you physically remove the
yeah, so we washed the roots with sterilized water. We didn't want to sterilize the root because, again, the plant were actually still in a tropical rainforest. We did not dug the root out, so we couldn't sterilize that. We only sterilized the water that we washed the root with, but this doesn't necessarily mean we got rid of all the microbial communities. We basically just got rid of what's in the surface that we were able to wash with water. So there should be some microbial communities left, but overall we should have decreased the abundance of microbial communities that are living on the roots.
So the nodules were removed.
We believe that it was probably harsh enough to remove some of the nodules. Yeah, especially what we observed with Clitoria. And it could be also that the roots that we use for Clitoria were actually did not have a nodule, but we also believe that as you remove, no matter how gentle you are, as you try to dig the roots out of the soil, you might have actually disrupted the root structure, and then having them within the tubes and then covering them back with soil, it might have actually disrupted some of the root structure and allowed us to lose some of the nodules, in addition to losing the nodule because of the washing.
Thank you. What can I say? An amazing story.
Thank you. Thanks, Dragus.
Okay, Amir.
Hey, Malak, great talk. I have a couple of questions if we have time, but the first one is, so you mentioned you collected soil or rhizosphere metabolite. I just want to first, clarify that. How did you collect that, and can you consider those as actual root exudates, or where those metabolites were located?
Yeah. So what we've done at the end of the experiment, we actually cut the roots, and then we shook the roots, and whatever soil come off the root, we consider that as rhizosphere soil. So that's why we believe it's directly influenced by the root exudates. So it's a combination of directly root exudates as well as some of the what's been in contact with other of the bulk soil matrix.
Thank you. And the other one is, so you did multi-imaging. I'm assuming that was not the whole root. So you choose a particular part of the whole root system or root zone?
Yeah, we did a transverse cross-section, so we could look at the root from top to look at the internal structure.
That was kind of in the middle of the root, or?
Yeah, so you know how we took we dug the roots out, and then we gave them pyruvate. The end of the root was actually getting the pyruvate, and then we cut the root in between, kind of like take a small cross section from where we're giving it the pyruvate all the way up. So whenever we got that cross section was randomly in between that space that we were actually measuring.
Okay.
So we did not discriminate between fine root versus non-fine root.
Thank you very much.
More? Oh, okay.
My question already been answered. Thanks. That's amazing talk. Yeah.
Thank you.
I had a question just the three-dimensional information, but I guess you already answered. Thanks. Yeah.
Right, no problem. Thank you.
Any other questions? Here we have
Adeta.
a data.
Thank you.
Data, wait for the microphone, just that online people can hear you.
Great talk, Malak, and I really enjoyed it. I have a quick question about the conditions that you selected, the drought conditions.
Mhm.
How extreme are those that you selected? Is it something you expect to happen, or is like the end member of the drought?
I think we went through a 2-month drought condition, which I think it's reasonable for tropical rainforest to go through that time. And when we looked at the moisture, we actually, by going through 2 months of drought, we dropped the moisture by 50%, and the temperature increased, the topsoil temperature increased by around 3 degrees. So the reason we chose 2 months, first of all, we want to see some changes going on, but I think it's reasonable within what tropical rainforests are expected to go through those drought conditions.
We have time for one more question, so. Peter.
Hey, Peter Zwart, Berkeley Lab. A fantastic story. I have a question about the variability in the metabolites among different plants and among different sections. So how big is that?
There is definitely variability, and I think we only used 3 replicates just because it's the feasibility of the experiment. So there's definitely variability, but I think when you look at different plant species, I think the effect of the plant species is stronger than the variability that we see from one plant to another. Also, something else to consider is the age of that plant. For these 3 plants we took for each plant species, they were relatively similar age and they were at a mature age, so they are not necessarily growing. So all of these we had to think about before selecting the plant, but there is definitely variability. And if I have to go back and redo the experiment, we would take at least 5 or 6 replicates within the plant to be able to better understand exactly how the metabolome is changing, because we took 3 plants and for some of them, one of the plant looked different than the other 2. So it was a little bit hard, but since we had 3 different complementary tools, it allows us to really validate our observation. But it would have been risky if we only use one analytical tool with only 3 replicates of plants, because definitely there's a huge variation between the plants, especially also where they are grown in the tropical rainforest and what's the surrounding environment, what kind of organic matter they're living on.
Yeah, biology is very complex.
Yeah.
One last question. Stress response when cutting, do you expect to see anything?
So we only cut at the end of the experiment and then we flash froze right away. So this way, if there is any, we wanted to, I mean, you have to cut the plant and we flash froze so that we don't have any metabolism happening. Of course it would, but I'm sure that wherever we collected the MALDI images were actually from within the middle of that root. So we avoided the edges of these root if there is any effect of the cutting of the root on the both ends of the root when we cut that.